Hello, everyone. My name is Gerold Feuer, and I'm the Senior Director of Scientific Research and Innovation at Crown Biosciences, a preclinical contract research organization providing high-quality models, technologies, and support for your drug development needs. Today's 15-minute webinar is a part of an educational tech talk series focused on in vivo solutions for oncology and immuno-oncology drug development. There will be no Q&A session at the end, but please feel free to ask questions in the chat box and we will answer them as quickly as possible. My talk today is entitled Humanized Mouse Models for IO Drug Development. So what we're going to cover today is the immune system's humanization strategies in creating humanized mouse platforms, the pros and cons of each humanized mouse model, and we're going to describe some case studies where we applied the humanized mouse models to test cancer immuno-oncology therapies. So the explosion of checkpoint inhibitors has really revolutionized cancer therapy. The first one being anti-PD-1. And what's really needed is predictive preclinical models for testing the efficacy and as well as the safety of some of these monoclonal antibodies. And the transgenic GEM and syngeneic mouse models have been the workhorses of immunotherapy research. However, they're not suited for testing human-specific immunotherapies. So there's a great need for creating humanized mouse platforms that are applicable in testing for the efficacy and the safety of these new immunotherapies. And the humanized mouse models that we employ at Crown are basically two types. One where we inject human PBMCs into mice. This is a very rapid humanization process. It takes about 2 to 3 weeks. And when you look at the mouse, when you look at the peripheral blood in these mice, you can actually detect human CD45 cells as shown in the flow diagram here, 2 to 3 weeks after engraftment. The other model that I'll describe in a little bit is basically injecting mice with human hematopoietic stem cells or CD34 cells. This process, you have to myoablate the mouse first to clear out the bone marrow, but what you have is human stem cell engraftment, hematopoietic stem cell engraftment in these mice, and you have a very long-term, robust humanization. And these mice, however, take 10 to 12 weeks to demonstrate their humanization potential. So both models have certain applicabilities and certain drawbacks. And here we list the pros and cons of each model. So the mixed xeno model is essentially the human PBMC model. Again, it's very rapid in humanization. It's relatively inexpensive because of that. And you can go back and actually do successive experiments using the same donor PBMC. So you have the same genetic background in generating these mice. However, there's some level of graft versus host disease, GVHD, depending on the strain. So the studies that we employ are relatively short in analysis, so before GVHD occurs. And there's quite a bit of allogeneic reactivity in these mice. In the human CD34 model, again, these are very long-term, stable, robust models that show human hematopoiesis. It's a little bit more of a de novo humanization in that you get stem cells that actually develop into the full range of human immune cells. The human T cells actually get educated on the mouse thymus. So there's not as much, there's very little graft versus host disease, depending on how we construct these mice. And there's also development of monocytes and NK cells in some of these models. The drawbacks of these are obviously it takes 10 to 12 weeks, so it's a much longer winded process. And these are more expensive than the mixed xeno models. However, both models can be used to evaluate checkpoint inhibitors. The mixed xeno mouse is additionally valuable in that you can check for cytokine release syndrome, which is a problem with certain monoclonal antibodies. But you can assess the efficacy of BiTE-like antibodies, ADCC monoclonal antibodies, CAR-Ts, efficacy, as well as NK modulating agents. In the human CD34 mouse, this has a broad range of applications in oncology, especially in evaluating IO drugs, including CAR T therapies. And it also has the ability to look at infectious diseases, hematopoiesis, and gene therapies as well. So the platform of the humanized mouse is particularly appealing because you can actually engraft either cancer stem cell lines or patient-derived samples. And especially the advent of the PDX models is very powerful in combining that with the humanized mouse platform. So these either cancer cell lines or PDXs will engraft in these humanized mouse because these mice are immune deficient. And this combination of engraftment in a humanized mouse allows you to then evaluate anti-tumor immunotherapeutic approaches. And I don't need to explain that these IO drugs have created a very revolutionary amount of, have really revolutionized cancer therapy. And not only in terms of their ability to work independently, but also to work in conjunction with traditional therapies and genomically targeted therapies. And these humanized mouse platforms are especially appealing in evaluating not only single therapies, but combo therapies. And this just kind of illustrates the advantages. So these humanized mouse platforms are particularly relevant in developing or evaluating human-specific antibodies where there's no mouse surrogate and there's no cross-reactivity to mouse receptors. There's these platforms also are relevant in evaluating novel immuno-oncology targets. So they could be used for essentially marker discovery, and then also in combination therapies. You can actually design experiments looking at the effects of combo immunotherapy treatments, which is more and more the standard operating procedure nowadays in cancer therapy. You can also combine immunotherapies with targeted agents, namely like driver mutations. And finally, these models can access the diversity across populations for both the immune system and for the tumor diversity, which is also a powerful appeal to these models. So I'm going to describe a mixed xeno mouse model case study. The mixed xeno models, again, are the PBMC engrafted mice. They do develop GVHD 2 to 3 weeks post-immune cell engraftment. So that shortens the experimental window that we can utilize. And these are best positioned to investigate immune cell reactivity against fast-growing tumors. So this case study explores an effective strategy to manage GVHD, using mixed xeno models and the type of immunological functions which can be investigated in these models. So this is an illustration of just how we kind of titrate the PBMCs and look at the CD45 expression levels in the blood at day 16 after injection. And we either inject them IP or IV. And as you can see, IP is a more efficient way to reconstitute these mice. However, you really want to titrate these PBMCs in to determine like the optimal level of engraftment. And why is that? Because you also need to synchronize, and in this case, it's the non-small cell lung carcinoma line. You need to synchronize the growth of the tumor alongside the PBMC human immune system reconstitution. So in this case, you see that we titrate in PBMCs, and we also evaluate and synchronize the growth of this tumor. And what we see is with this arrow pointing at the red line is that 5 million PBMCs injected IP is actually an ideal amount of cells injected for this tumor cell line. So again, we've done a lot of these case studies. We have experience looking at these engraftments and synchronizing tumor cell lines in order to really set up the model for testing IO drugs. And this is exactly an illustration of this. This is looking at lung cancer cell line and looking at an anti-PD-L1 antibody effect. So this is in humanized mouse, engrafted with the lung cancer cell line, and looking at anti-PD-L1 efficacy. What we can see in this graph is that if we look at a mixed xeno model engrafted with a lung cancer cell line and test these mice for anti-PD-L1 injection, what we can see is tumor graft inhibition in the lower line here in red is much more substantial when these mice are treated with anti-PD-L1. But importantly, the PBMC has to be present, the humanization has to be present for these IO drugs to work, for this anti-PD-L1 to work. And if we look at the T cells, CD3 T cells, the CD3, CD8 effector cells, and CD4 cells, what we can see is there's an elevation of T cells in the mice treated with anti-PD-L1. And furthermore, there's an elevation of the TILs that are demonstrated in these animals. So again, the anti-PD-L1 treatment has a similar effect, reflecting what happens in humans. So the conclusions is that GVHD is an undesirable but manageable side effect of PBMC engraftment in these humanized mixed xeno mice. We can use this platform to evaluate the anti-tumor efficacy of IO agents in vivo. We can also target T and NK cell functions in these mice. And optimal inoculation protocols for both immune cell and the cancer cells need to be done for each model. And we have a certain number of models that we've already validated in these mice. Furthermore, we've just licensed in NSG murine MHC-1/2 knockout model, and these show significantly reduced GVHD, so that we're fairly confident that these mice will eliminate a lot of the protocols that we need to manipulate in order to set up these experiments. So there's a lot of promise for these mice in the future. So I'd like to switch gears to the human CD34 mouse models. And on the right here, again, these mice, we myeloablate them, then we inject them with human CD34 hematopoietic stem cells. And 10 to 12 weeks, you see significant humanization in the peripheral blood of these animals. And if there's two strains of mice that we employ, the NSG mouse and as well as the SGM3 mouse. And if you look across both of these, they're very similar. The SGM3 mouse actually has transgenic expression of human IL-3 and GM-CSF. And what we can see is that there's significant humanizations in either strain, in terms of this human CD45, the T cells, the human CD19 B cells. But moreover, you also, especially in the SGM3 strain, you have development of monocytes, macrophages at 10 to 12 weeks. So that's an additional advantage of these animals, is that we can investigate the effects of macrophages in combination with these therapies. So again, immunotherapies have shown unprecedented results in patient survival. However, some patients do not respond to current immunotherapies. These preclinical humanized mouse models are necessary for testing synergistic effects and evaluating combination effects. But we also use these models to look at engraftment of PDX and CDX xenografts in either of these strains. And what we've developed is basically a checkpoint inhibitor benchmarking study, what we call a humanized mouse clinical trial, which allows the evaluation of the variations of the donor and the tumor response to immunotherapies. So I'd like to present a case study actually demonstrating this. So what we have in this case study is humanized tumor-bearing models to predict the response to IO therapies. And we design it in a checkerboard study design with an N number of 1, meaning that we use multiple immune cell donors and multiple lung cancer models and graft each of those into one animal. and test the IO drug therapies on these. So we screened 30 different lung cancer PDX models for PD-L1 expression, and just chose the high-level expression PD-L1 expressing models to engraft into these mice. And what we see here is basically the mouse humanized mouse clinical trial for IO drug development. And what you have on the y-axis is a number of different donors, so 5 different human stem cell donors that were used to create humanized mouse. And on the x-axis are 7 different lung cancer PDX models that all express high levels of PD-L1. And then each one is engrafted into one mouse, and all of these mice are then treated with Keytruda. And what you see on the right is basically the tumor growth inhibition, a waterfall plot. And basically what you see in the blue lines is that about 36% of the models that are engrafted actually show a response to Keytruda. And importantly, this reflects the patient variability to Keytruda responsiveness. So the combination of the genetic background of the donor alongside with the genetic background of the tumor reflects the variability in the responsiveness of this IO drug. And then if we actually go into these mice that are responders versus non-responders, it's very interesting, because if we look at the number of TILs, actually, they're actually increased in the responders. So again, this reflects what happens in patients, that the responders have an increased infiltration of TIL T cells into the tumor itself. But more interestingly is actually that the macrophages, which again do develop in these humanized mice, the macrophages in the responders are actually classically M1 classically activated macrophages, and the non-responders show overrepresentation of M2 macrophages. So again, these mice actually do reflect what happens in actual patients in terms of the human immune system and the types of human cells that infiltrate the tumor. So the variability of the IO response depends on the immunological state of the test subject. The IO models and study designs should reflect this variability. And these human CD34 humanized mouse models engrafted with PDX are predictive of responses observed in the clinic. So we think these are very valuable models to help not only to test therapies, but also to help guide biomarker discovery and patient selection. So to summarize, the models with functional human immunity can be used to test for the safety and efficacy of these IO drugs. The specific questions of the protocol or the study that is being addressed should really guide the model selection and the study design. And we at Crown Biosciences are here to really help you decipher out exactly the proper model and the proper outcome that should be employed for these model designs. Finally, understanding the immunological features and limitations of each model are really the keys to success. So we'd like to thank everyone for participating in today's Tech Talk. I hope you enjoyed it. And please join us for the next talk in the series titled Predicting Clinical Response with Mouse Clinical Trials by my colleague, Dr. Shen Gao. To learn more about the relevant resources, visit the resources section of the video platform and visit us at crownbio.com. Thank you.